Okay, so I did my topic on brain matter as a toxicological specimen. So to start it off, I want to talk about something that is vital in the protection of the brain. All organisms that have a well-developed central nervous system have something that is called a blood-brain barrier. This develops during fetal life and is well formed by the time of birth. The blood-brain barrier consists of endothelial cells that can form the walls of the capillaries, and it prevents many macromolecules from entering the brain by serving as a protective shield from neurotoxic substances that circulate within the blood. An example of neurotoxic substances that I will be talking about specifically can include drugs. So the tight functions are a key feature of the blood-brain barrier, and they are illustrated right here by the black squares. So these significantly reduce permeation of polar solutes through diffusion between the endothelial cells and the blood plasma to the brain matter. So these are responsible for the restriction of polar solutes and macromolecules by blocking penetration. Something that is essential for the formation of these tight junctions are something called adherence junctions that are present along with the tight functions. So they hold the cells together by giving the tissue structural support, and the blood-brain barrier is maintained through the formation of these tight junctions. So there are a few ways that substances are able to be transported across the blood-brain barrier. The first way that I'll talk about is through passive transport into the brain. These include things that are lipid soluble that can enter the brain passively. Some things that cannot enter the blood-brain barrier passively include things with a high polar surface area, the formation of more than 6 hydrogen bonds, presence of rotatable bonds, or a molecular weight that is greater than 450 daltons. Another way that something is able to transport across the blood-brain barrier are through solute carriers. So solute carriers, or transporters, as they are also called, supply the central nervous system with polar nutrients like glucose and amino acids that are needed within the body. The third way are through ATP binding cassette transporters, or ABC transporters. ABC transporters are a family of proteins. Their major role is to function as active efflux pumps that consume ATP and transport a diverse range of lipid-soluble compounds out of the brain, endothelium, and the central nervous system. So they carry out neuroprotective and detoxifying functions by removing potentially neurotoxic drugs from the brain. The transport of macromolecules across the blood-brain barrier is another way. So endocytotic vesicles are the main route for any solutes that enter the brain that have a large molecular weight or are particularly heavy. So I think the cutoff was about 450 daltons. Macromolecule ligands bind to specific receptors on the cell surface, and this triggers something that is called an endocytotic event. The receptors and the bound molecule are able to pinch off into its own vesicle and go across the cytoplasm. This is to be exocytosed at the opposite end of the cell. The ligand then dissociates from the receptor and then is within the brain. Cell movement is the final transport across the blood-brain barrier. So brain penetration occurs only after traumatic events. This is when activated neurophiles damage the blood-brain barrier. So this could include any traumatic event to the body that leads to the deterioration of the brain, and thus it allows for cells to move within the brain. So drugs and the blood-brain barrier. Psychoactive drugs can alter the permeability of the blood-brain barrier. Methamphetamine and cocaine have been shown to induce changes in the blood-brain barrier specifically. Methamphetamine and cocaine increase oxidative stress. This then produces something called reactive oxygen species, or ROS. Oxidative stress causes vascular endothelial dysfunction because it increases permeability and promotes adhesion or inflammation. When reactive oxygen species are created, it allows for the movement of monocytes, which are white blood cells. They lead to the rearrangement of the tight junctions, which are so vital to the blood-brain barrier, and this reduces the protective nature of the brain. Methamphetamine was shown to degenerate neurons in both the hippocampus and the amygdala. Drugs of abuse increase temperature within the body, so this also affects the permeability of the brain and the thermotolerance of the blood-brain barrier. This elevated brain temperature is produced from drugs of abuse and it breaks down the blood-brain barrier. So postmortem toxicology is relevant to this topic of using brain as a biological specimen. This subdiscipline is relevant to brain matter because brain matter can only be collected in a postmortem case. Postmortem toxicology is defined as determines the absence or presence of drugs and their metabolites, chemicals such as ethanol and other volatile substances, carbon monoxide and other gases, metals, and other toxic chemicals in human fluids and tissues, and evaluates their role as a determinant or a contributory factor in the cause and the manner of death. So using brain matter as a toxicological specimen has not been very widely used, but it is useful in some specific cases. Something if a traumatic injury occurred that led to a significant blood loss or say contamination of the blood from a ruptured stomach, then brain matter would be an excellent choice of a specimen to use. Research has shown that there are positive trends and correlations that are observed within the brain tissue, and research done has provided insight into using brain as a toxicological specimen and as another tool in order for toxicologists to determine drug concentrations post-mortem and be able to compare them throughout the body. So in a study done by J.B. Dietrich, they analyzed how the blood-brain barrier can be altered by drugs of abuse. They specifically looked at methamphetamine and cocaine, which I talked a little bit about earlier. So this approach was to evaluate the measured drug concentrations in blood and compare that concentration to the concentration of the drug within the brain. The method of collection was to gather brain tissue from the cerebrum of decedents. The quantitative data was collected through the use of GCMS. The key results of this experiment included that opioids tended to have a heart blood concentration that was 1/2 of the concentration that is within the brain. Cocaine and cocaine metabolites had very similar heart, blood, and brain concentration values. And then we can remember from lecture that volume of distribution is defined as the theoretical volume of body fluids in which the amount of drug in the body is distributed at a concentration that is equal to that of plasma. And then we can also recall that post-mortem redistribution, or PMR, is the passive drug release from drug reservoirs. This can include anything from like the GI tract, liver, lungs, myocardium, and this occurs directly after death. So in this study in particular, it was concluded that with a high volume of distribution, a high PMR is expected. This meant a low ratio of heart blood to brain matter. Some examples of drugs that showed this trend included dextromethorphan, promethazine, amphetamine, fentanyl, methamphetamine, diphenhydramine, and more. The opposite of that trend is that with a low volume of distribution, a low PMR is expected. This means a higher ratio of heart blood to brain matter. And the only drug observed doing this in this study was zolpidem, which is a sedative. And they acknowledged that morphine was one exception to this trend. So morphine has a high volume of distribution, but also a high heart blood to brain matter ratio. So this kind of goes across the two trends that I just explained. If you think about why this could be, it could be because morphine is extremely polar, meaning that its ability to penetrate the blood-brain barrier is a lot more difficult than other solutes. And then Using brain matter can be viable in some cases, while in others it may not. So, toxicologists should keep in mind the use of brain matter as a toxicological specimen because it could, in some cases, be the only available specimen or one of the only available specimens. So, some disadvantages of using brain matter include the fact that postmortem redistribution may alter the drug concentration when compared with the antemortem concentration. This is due to the passive diffusion of a drug from the high concentration tissues. Another disadvantage of using brain matter as a toxicological specimen is because it can be used in postmortem cases only, so it kind of limits your use. And some advantages of using brain matter as a specimen is that it is anatomically isolated. The brain also has less putrefaction or decomposition that occurs within it, so you have kind of a little bit of a longer window. And this also has to deal with the lower metabolic activity of the brain. This allows to be advantageous because it could have a longer window of detection for some drugs. And then the brain also undergoes less PMR, so the brain concentration is found to be a little more stable over time when compared to other sites of samples. And then here's some references that I used. So it included the study that I talked a little bit about, and then I also used our toxicology PowerPoints as well.